Hey man, have you heard about CRISPR?
CRISPR? What's that?
So CRISPR is this new genome editing technology. Here, have a look.
CRISPR-Cas9.
Mutations in these
Okay, so basically the way it works is when a virus infects itself into a bacteria, it'll inject its DNA straight into that cell. In response to this, the bacteria will create two strands of RNA, collectively known as the guide RNA, which form a complex with the Cas protein. Think of it like this. The guide RNA is the targeting system to the Cas protein, which is the warhead to make the incision in this viral DNA. So the guide RNA will exactly match the viral DNA down to the nucleotide, telling the Cas protein to make that double-strand incision in the DNA, thus destroying it. Bacteria then stores this DNA in a section of its genome known as the CRISPR locus. So what we have done to harness this process is tailor our own guide RNAs. So we can get the CRISPR-Cas9 enzyme to target anywhere on the genome that we want. And then by directing the route of cell repair, either by non-homologous end joining or homologous repair, we can insert or remove DNA anywhere on the genome. The potential is enormous.
Today we have Eva that will be talk to us more.
The properties of CRISPR allowing it to edit genetic material have made major advancements in disease prevention and genetic defect correction. One study to note revealed further HIV infection can be prevented using CRISPR editing technology through the eradication of HIV-1 from affected cells and the prevention of further HIV DNA replication. In transgenic mice spliced with HIV-infected human immune cells, after one application of CRISPR, all the viral segments of the human immune cells in the mice were removed. Another application of CRISPR can be seen through a recent study in China, where genetic defects in human embryos were corrected using CRISPR. This study was the first study to show that CRISPR editing technology is viable in human embryos. The study itself used immature eggs that were fertilized with sperm from either of two males who both had genetic disorders. In the eggs that were fertilized by the first individual, two embryos successively had the gene corrected, but a mosaic occurred in the other embryo. Mosaics occur when mutated genes are corrected in some, but not all of the cells of the embryo. In the four eggs fertilized by the second individual, the mutation was corrected in one embryo, a mosaic occurred in another, and the editing was unsuccessful in the remaining two embryos.
CRISPR significantly improves efficiency over previous techniques. Generating a knockout organism through this process could take in excess of 1 year. A knockout organism contains only the altered gene, or contains chromosomes where the gene has been removed. It is homozygous. Previously, a knockout organism, often a mouse, took multiple generations through inbreeding to achieve. The first generation mouse has an altered gene implanted into the embryo. The resulting hybrid has only a chance to produce an offspring carrying the altered gene. Subsequent generations are then inbred until a homozygous individual is produced. By contrast, CRISPR edits both copies of the gene simultaneously, and when used on stem cells, can reduce the chance of the initial mosaic expression, producing a knockout in the first generation.
Now the pressure's on.
Genome editing techniques such as CRISPR have opened up a plethora of opportunities for eradicating disease and life-changing genetic defects, potentially changing the lives of millions of people. However, these opportunities will, in turn, open a number of doors to serious ethical implications that cannot be reversed. Firstly, the introduction of CRISPR technology highlights some concerning questions regarding the equality and justice behind the technique, namely who will be allowed and able to access this technology and at what cost. Limiting the availability of this innovative technology to only individuals who can afford it creates a deeply unethical system, leaving those who cannot afford the treatment to suffer from what could be an easily curable disease. With designer babies already a reality in the modern world, CRISPR may create a pathway to irreversible changes to the human gene pool. In the near future, CRISPR is predicted to be a revolutionary technique that will allow parents to screen and remove potentially deadly or life-changing diseases that may affect their offspring. Diseases such as Down syndrome may be completely eradicated from the human genome. While this initially will drastically improve the quality of life for parents and their offspring, this sort of genetic alteration creates a slippery slope for what could become highly selective eugenics, potentially diminishing variation within the human population and thus creating unknown and potentially deadly mutations that we cannot treat. Conversely, some scientists and notable figures have highlighted the reverse side of the argument, highlighting that choosing not to use readily available genetic modification techniques that can cure disease or birth defects is unethical in itself, as it condemns an unborn child or innocent person to a life of difficulty, suffering, or even death.
Well, CRISPR sure is interesting, hey?
Yeah, this could have global implications.
Hey, where'd everybody go?